Hi, we are team number 4. My name is Jimena and we are gonna talk about the photosynthesis. First of all, what is photosynthesis? It is a chemical reaction that takes place inside a plant, producing food for the plant to survive. We can say that it is the formation of chemical energy stored in the glucose bonds and oxygen releasing in the presence of sunlight. Then on this slide we have the steps of the photosynthesis in the diagram. The first one is the absorption of light. This is through chlorophylls that are attached to the proteins in the thylakoids of the same chloroplast. Then the light energy absorbed is used to remove electrons from an electron donor. Then the electrons are further transferred to a primary electron acceptor, the quinine, which is similar to COQ in the electron transfer chain. The second step is electron transfer. The electrons are now further transferred from the primary electron acceptor through a chain of electron transfer molecules present in the thylakoid membrane to the final electron acceptor. The third step is the generation of ATP. The movement of protons from the thylakoid lumen to the stroma through the ATP synthase results in the generation of ATP from ADP and PI. The fourth and last step is the carbon fixation. The NADP and ATP generated in steps 2 and 3 provide energy, and the electrons drive the process of reducing carbon into 6-carbon sugar molecules. The first 3 steps of photosynthesis are directly dependent on light energy and are called light reactions. However, the reactions in this step are independent of light and are called dark reactions.
Now I'm going to explain the key names for proteins used in the photosynthesis process. We have 4 main proteins that are photosystem 1 and 2, ATP synthase, and cytochrome B6F. A chain of chlorophyll, phylloquinone, and 3 iron sulfur clusters are cofactors that convert the energy from light into energy that the cell can use. This electron, moved by the energy, is used to perform tasks such as the production of sugar from the carbon dioxide. Light is captured by the photosystem 1 for the process of photosynthesis. In the case of organisms with oxygenic photosynthesis, they have 2 kinds of photosystem, the photosystem 2 and the photosystem 1, which have as main photosynthetic pigment the molecule of chlorophyll. On the other hand, photosystem 2 is the first link in the chain of photosynthesis. It captures photons and uses the energy to extract electrons from water molecules. Then they pass down with a chain that is carrying proteins, and it moves along the way of photosystem 1. As these electrons flow down the chain, they are used to pump hydrogen ions across the membrane, providing even more power to the ATP synthesis. Finally, the electrons are placed on a carrier molecule, NADPH, which delivers them to enzymes that build sugar from water and carbon dioxide. These electrons are used in several ways. First, when the electrons are removed, the water molecule is broken into oxygen gas, and this forms ATP synthesis. That is the source of all the oxygen that we breathe. Finally, we have the cytochrome B6F. Its function is to mediate the transfer of electrons between the 2 photosynthetic reaction centers, from photosystem 2 to photosystem 1, while transferring protons from the chloroplast stroma across the thicaloid membrane into the lumen. Then we have the ATP synthase. That is a complex process which makes use of the proton potential created by the action of the electron transport chain in the mitochondria. It transports a proton down the gradient and uses the energy to complete the phosphorylation of ADP to ATP.
Okay, I'm talking about key names for enzymes used. The number 1 is lipases. It's a set of enzymes that acid digest fat inside the gut. Number 2 is amylase anabolase changes into sugars. Amylase is observed in saliva. Maltase additionally observed in saliva breaks the sugar maltose into glucose. Maltose is observed in ingredients which includes potatoes, pasta, and beer. The number 4 is trypsin. Observed inside the small intestine breaks proteins down into amino acids. The number 5 is lactase, additionally observed when inside the small intestine breaks mirror acetylcholine in nerves and muscles. The number 6 is helicase. In reduce is unravels DNA. And the last is DNA polymerase. Synthesis DNA from deoxyribonucleotides, lactose, the sugar in milk into glucose, and galactose.
Sunlight and types of wavelengths. What is a sunlight? Well, a sunlight is basically a set of waves with different spectrums. There are different types of radiation and sunlight that are going to be mentioned right now. The first type is called the ultraviolet rays A, B, and C. These are the type of rays that cannot be seen by the human eye and by which have various properties, such as sterilizers because they can eliminate germs. The second type is the visible range. This is the type of light the human eye can catch, and this has different colors according to the wavelength in its time and space. And the third type is the infrared range. This type of radiation is responsible mostly for the heat provided by the sun, impacting our Earth.
There are two stages or reactions of photosynthesis, the light-dependent reactions and the light-independent reactions. In the light-dependent reactions, the energy from sunlight is absorbed by chlorophyll and it is converted into chemical energy in the form of electron carrier molecules like our ATP and NADPH. These light reactions take place in the thylakoid membranes of organelles, which are called chloroplasts. In the light-independent reactions, the energized electrons from the light-dependent reactions provide the energy to form carbohydrates for the synthesis of glucose. These light-independent reactions are sometimes called the Calvin cycle because of the cyclical nature of the process.
Adenosine triphosphate, also known as ATP, can be defined as a fundamental nucleotide in obtaining solar energy. It is found in the cells of all living things. It captures chemical energy obtained from the breakdown of food molecules and releases it to fuel other cellular processes. Amount of ATP used in photosynthesis. There are different amounts of ATP that are produced, depending on the process it's in or the phase in which it's running. During the light phase, 18 ATP is produced. In glycolysis, 8 ATP is obtained, and in the dark phase, when there is no light, there is no ATP production.
The second topic that we are going to talk is the cellular respiration. First of all, what is cellular respiration? It's the process through which cells convert sugars into energy to create ATP and other forms of energy to power cellular reactions. Cells require fuel and an electron acceptor which drives the chemical process of turning energy into a usable form. On this slide, you can see that we have two different types of cellular respiration. The first one is the aerobic. This one uses oxygen to turn fuel into chemical energy. It is more efficient and produces ATP quicker than the anaerobic respiration. This is because oxygen is an excellent electron acceptor for the chemical reactions involved in generating ATP, as we can see on the diagram. Then, on the right side, we can see the anaerobic respiration diagram. This is the type of respiration through which cells can break down sugars to generate energy in the absence of oxygen. This is the main difference between both. As I already said, the aerobic respiration is better because it is more efficient and quicker, but this one helps the organisms that can perform respiration without oxygen to perform it. But they also have similarities, which are that they both are methods of harvesting energy from a food source, such as fats or sugars, and both processes begin with the splitting of a 6-carbon sugar molecule into two 3-carbon pyruvate molecules, and this process is called the glycolysis.
Okay, so enzymes. Enzymes are proteins that catalyze or have an effect on the rate of chemical reaction without themselves being altered inside the method. Specific enzymes catalyze a hemimobile response. Glycolysis, the first step of the respiratory responses, takes place inside the cytoplasm or fluid of the mobile. Glycolysis includes 9 separate chemical reactions, every catalyzed with the aid of using a particular enzyme. The citric acid cycle. The second step of the respiratory response takes area internal a mobile organelle referred to as the mitochondria, which, because of their position in ATP manufacturing, are referred to as electricity factories for the mobile. And so, specific enzymes positioned within inside the mitochondria that electricity the various reactions that make up the citric acid cycle. And oxidative phosphorylation, the very last step of the respiratory response, additionally referred to as the electron shipping chain, is wherein the strength payoff takes place for the mobile. During this step, oxygen drives a series of electron motion throughout the membrane of the mitochondria. This switch of electron powers the potential of the enzyme ATP synthase to supply to 38 molecules of ATP.
Electron transport and ATP synthesis. This is the last stage of the cellular respiration. In this last stage, it is called the electron transport chain. Electron transport. High energy electrons from NADH and FADH2 are passed from carrier to carrier down the electron transport chain. Water is formed when oxygen accepts the electrons in combination with hydrogen ions. Energy generated by the electron transport chain is used to move H+ ions across the inner mitochondrial membrane and into the intermembrane space. And the ATP production is when H+ ions pass back across the mitochondrial membrane through ATP synthase, causing the base of the synthase molecule to rotate. With each rotation driven by the movement of the H+ ion, ATP synthase and attaches a phosphate to ADP to make ATP.
There are 4 stages of cellular respiration. The first one is glycolysis. In glycolysis, glucose, which is a 6-carbon sugar, undergoes a series of chemical transformations. In the end, it gets converted into 2 molecules of pyruvate, which is a 3-carbon organic molecule. In these reactions, ATP is made and NAD+ is converted into NADH. The second one is pyruvate oxidation. In this one, each pyruvate from glycolysis goes into the mitochondrial matrix. There, it is converted into a 2-carbon molecule bound to coenzyme A, known as acetyl-CoA. Carbon dioxide is released and NADH is made. The third one is citric acid cycle. In this one, the acetyl-CoA, made in the last step, combines with a 4-carbon molecule, and it goes through a cycle of reactions, ultimately regenerating the 4-carbon starting molecule. ATP, NADH, and FADH2 are made and carbon dioxide is released. And the last one, which is oxidative phosphorylation. The NADH and FADH2 made in the other steps deposit their electrons in the electron transport chain, turning back into their empty forms, which were NAD+ and FAD. As electrons move down the chain, energy is released and it is used to pump protons out of the matrix, forming a gradient. Protons flow back into the matrix through an enzyme called ATP synthase, making ATP. At the end of the electron transport chain, oxygen accepts electrons and it takes up protons in order to form water.
Amount of ATP used in cellular respiration. Cellular respiration is divided into two, aerobic and anaerobic. In the first one, there is a presence of oxygen, while in the second, not. Aerobic cellular respiration. This process occurs in the mitochondria in two stages called the Krebs cycle and the oxidative phosphorylation. A total of 38 ATP is produced in aerobic respiration. Anaerobic cellular respiration. This mechanism only produces 2 ATP molecules.
The third topic that we are going to talk is the fermentation. What is fermentation? This is a chemical process by which molecules such as glucose are broken down anaerobically. There are two types of fermentation, alcohol fermentation and lactic acid fermentation. On this slide, we can see the processing diagrams of the two types of the fermentation. First, in alcohol fermentation, the pyruvate from glycolysis is acted upon by the enzyme pyruvate decarboxylase, converting it into acetaldehyde and liberating CO2. The acetaldehyde is then reduced by the NADH to ethanol, and in doing so, NAD+ is regenerated to oxidize more glucose from substrate-level phosphorylation in glycolysis. And on the other side, we have the lactic acid fermentation. The pyruvate is directly reduced by the NADH to lactate, and that's all the process.
Key concepts of the fermentation. Alcoholic fermentation, under this demonization, is the system of giving the yeast anaerobic strength with the advanced oxygen and glucose. Lactic fermentation. In the case, it must be said that the microorganisms in and glucose play an essential role in it. And the main ones are the people who carry out this system, in which lactose is oxidized in part to get strength. A clear example of this inactivity in the acidification of milk. And acetic fermentation. In this different case, what happens is the cardiovascular microorganisms are responsible for remodeling the known as ethyl alcohol in what is acetic acid. And the last one is butyric fermentation. This different system must be cited as the only one in which, in the absence of oxygen, the microorganism Clostridium butyricum, the amylobacteria, converts where are carbohydrates into butyric acid.
We have three main types of fermentation: the lactic, the alcoholic, and the acetic acid. In the lactic acid fermentation, yeasts and bacteria convert starches or sugar into lactic acid in foods like yogurt, bread, and pickles. Then we have the alcoholic fermentation, where the pyruvate molecules of sugar are broken down by yeast into alcohol and carbon dioxide molecules to produce wine and beer. In the last type of fermentation, we have that the sucrose, that is the natural sugar of fruits, and glucose oxidate and break into the sour starches to form condiments and vinegars.
Now I'm going to talk about some main points of fermentation. Glycolysis. Glycolysis is the first step in the breakdown of glucose to extract energy for cellular metabolism. 6 carbon sugars are oxidized and broken down into pyruvate molecules. The corresponding anabolic pathway by which glucose is synthesized is called gluconeogenesis. Aerobic glycolysis, it is a series of reactions where oxygen is required to reoxidize NADH to NAD+, hence the name. This 10-step process begins with a molecule of glucose and it ends up with 2 molecules of pyruvate. Now, pyruvate is the end product of glycolysis. It is converted into acetyl-CoA and enters the Krebs cycle only if there is enough oxygen available. When the oxygen is insufficient, pyruvate is broken down anaerobically, creating lactate in animals and ethanol in plants.
energy, and exercise. Basically, at each stage of the race, your body uses different paths to get energy. You have three main sources of ATP: ATP already in the muscles, ATP made by lactic acid fermentation, and ATP made by cellular respiration. At the beginning of a race, the body uses all three sources. However, stored ATP and lactic acid fermentation can give energy only for a short time. The two types of energies are quick energy and long-term energy that are going to be explained forwardly.
Amount of ATP used in fermentation. Fermentation is another anaerobic way to break down glucose. Both fermentation and cellular respiration begin in the same way, with glycolysis. The energy obtained from pyruvate was produced in glycolysis. 2 ATP molecules are produced.
As a conclusion, we'd like to mention that throughout this stage, we learned a lot about photosynthesis, cellular respiration, and fermentation, about their stages and steps and characteristics, and their differences between one another, and how each one of them is really, really important and necessary. Thank you.